Catechol oxidase activity. In this experiment, we measure the performance of catechol oxidase to establish a baseline for temperature and pH experiments. An introduction to the experiment. The enzyme is catechol oxidase. Its function is to oxidize catechol. The enzyme creates a brown pigment when oxygen is present. So the reaction is catechol and oxygen become benzoquinone and water, and this reaction is carried out by the enzyme catechol oxidase. In this experiment, the positive control is tube 1. This tube contains the enzyme catechol oxidase, the substrate catechol, and oxygen, which is required for this reaction. The negative control is tube 2 because it contains no substrate, and the experimental tube is tube 3. We're trying to figure out if an enzyme helps the reaction. The procedure was to label 3 test tubes. We pipetted 1 ml of catechol and 1 ml of catechol oxidase in tube 1. As you can see, there's the substrate, the enzyme, and then oxygen is in air that we breathe, so that was there too. We pipetted 1 ml of water and catechol oxidase in tube 2. There's no substrate here. We pipetted 1 ml of catechol and 1 ml of water in tube 3. There's no enzyme in this case. So we were trying to see, does an enzyme really speed up the reaction, or can this reaction occur efficiently without an enzyme? And then, to carry out this test, we placed the test tubes in a 37°C water bath for 10 minutes. And we recorded the results using the scale that you provided on the wiki page. So here's a picture of our test tubes. Test tube 1 here is the positive control, and test tube 2 is the negative control, and this was our test tube 3 was our experiment. So as you can see here, the potato extract that we used was a brown color, hence that this is a little brown, as you can see over here. And when after the water bath, this is what it looked like. It turned a little darker. It's hard to see in this picture because of the table sort of makes, and the lighting makes this look darker. And the negative control stayed the same. There was no substrate to react with, which is what we expected. And based on this test tube 3, we realized that an enzyme is needed for this reaction to occur efficiently because there was no change without the enzyme. So tube 1 had a color change of about on a range of 2. The color is brown, which is proof of reaction. So the brown coloring is caused by benzoquinone, which we talked about before is the product of this reaction. And this is formed to protect the cells, or the fruit cells, plant cells, from the bacteria. Tube 2, which we expected to be our negative control, had a color change of 0, which was good. The substrate was not present in the scenario, so the enzyme does not bind to anything, and there's no production of benzoquinone. In test tube 3, there was also no reaction. The enzyme is not present in this reaction, hence there was no color change. Because the enzyme is not there, the activation energy required to get over the hump, the initial investment, that never took place. So catechol and oxygen were not converted into benzoquinone and water. Our conclusion is catechol oxidase is needed for the reaction to occur efficiently. Without the enzyme, as shown in tube 3, the reaction did not take place within 10 minutes, and 10 minutes was how long we left it. We left the tube in the water bath at 37 degrees. And catechol oxidase oxidizes catechol in the presence of oxygen, as seen in our positive control. So these two conclusions we'll be using in our future experiments in this lab, which were the temperature experiment and our pH experiment.